So far in this class, we've been really looking at how biology and all of life is built from the ground up. And we've discussed how the building blocks of all the big proteins and DNA are built from atoms. Last time we talked about how membranes are made up of little lipids. And today we're going to talk about once you get those membranes and those little enclosures, how do they build up larger organelles? And we're just going to kind of finish up some of the reasons for having boundaries today and talk a little bit more about that interesting outboard motor, the bacteria flagellum. So last time we talked about how boundaries are important for creating the driving force, getting all the ions on one side and not on the other. And so the rushing through the membrane, which can only happen in controlled fashions when the cell opens up a little door and lets them through, that powers a lot of the things inside of the cell. It creates ATP, which is the currency for all of life's activities. Well, that's not the only reason for boundaries. Boundaries, as we mentioned, are also to help put larger cells into bite-sized areas and to keep incompatible chemical reactions from happening right next to each other. All right? Well, why is it that cells that are large need to divide themselves up into bite-sized areas? Well, in a small cell like a bacterium, every protein can touch every other protein every second, which is pretty amazing when you start to look at pictures of how crowded it is and how many proteins there are. It's a giant city. But because it's so small, and the average speed, even through liquid, of a protein is very high compared to the size of the bacterium, everything can touch everything else every second. It's just all being mixed around. Now, this would not work in larger cells. Nerve cells, for example, can stretch from the small of your back to the tip of your toe. In a giraffe or in a blue whale, those neurons can be meters long. And if you were to wait for a protein to touch another protein on opposite ends of that neuron, I calculated it would take about 8,000 years. So that's clearly not going to work. Instead, those larger cells have to be divided into smaller bacterial-sized enclosures so that within that enclosure, everything can talk to everything else every second. And then if two enclosures need to encounter one another to do some kind of interactions, then there are motors, like the kinesin motor we've been talking about in this class, that walk and can pull behind them these enclosures. So that works really well for larger cells, and that keeps everything interacting. A good way to think about this is a little mental activity you can do. Imagine having a bowl of soup, clear broth, stirring it and letting it cool down, or just letting it sit on the table as the steam comes off of it. And imagine right next to it, equally hot, same size, is a bowl of oatmeal. Which one do you think is going to cool off faster? So if you come back in 15 minutes and dig right into the center and stuff it in your mouth, which one is going to burn your tongue? It's pretty obvious that the oatmeal is going to stay hotter longer. And if you don't believe me, you can try this yourself. So why is it that the oatmeal didn't cool down as fast as the clear broth or the cup of tea that's the same size? Well, it's because the cup of tea or the broth can have the little molecules of water moving around. So a hot one in the center can move to the surface and release the heat, evaporate, send molecules off that help evaporate, radiate the heat away. Whereas in the oatmeal, everything is sort of stuck. Molecules of water can't travel through as quickly. And this relates to proteins moving around inside the cell, because as the cell gets thicker, as there's more and more crowding inside, things don't move as fast. Chunks of oatmeal can't move up to the top, and that also illustrates that if you do have an enclosure, which is great for everything inside of the enclosure, then there becomes a real problem with moving that enclosure because it's even bigger, so it moves even slower than a protein would. And in fact, I calculated that a normal size enclosure that holds some kind of reactions would take orders of magnitude longer than the age of the universe to move from one side of our nerves to another. So it gets really to be a problem. And without molecular motors like kinesin, this would, of course, never happen. So that's a good mental activity for you to think about. Now, let's return to the story we were talking about earlier about the bacteria flagellum. We know that there's this little corkscrew that sticks off on the end of a bacterium, and it's made of lots of fibers that come from different outboard motors. And when they're all working counterclockwise, they all bundle up really nicely, and it looks like a single corkscrew, just like the kind you drive into a cork and pull out, just exactly that same shape, that helical shape. But when it's going clockwise, it flies out into all different directions. Now, this is important, actually, for helping the bacteria find its food and avoid toxins. When the bacteria senses that food is becoming more and more plentiful, then it sends a signal to all of its outboard motors to continue spinning counterclockwise. And that keeps the bacteria moving in the same direction, which is probably a good direction since food is getting more plentiful. If food is getting more scarce as it's traveling along, it realizes, well, maybe I might be going in the wrong direction, and so it sends a signal to go clockwise. All those same little fibers go all over the place. The bacteria starts tumbling, and it finds a new direction, and so when it goes back to being counterclockwise, the bacteria might jet off in a new direction. Now, that is an example of how to keep things moving even at the molecular level. And so what we've been talking about today has to do with diffusion, has to do with things moving around, and we'll be getting into this a little bit more in the next few lectures. Now, if you're looking at the textbook, BioBook, there's a branch that you'll want to look at and all the leaves associated with it. The branch is called What Are the Major Compartments, Structures, and Spaces Inside of a Cell? And what I recommend is it has a list of subcellular components. You can click on each one of those for a short definition. It'll pop up a little definition if you don't know what it is. And the rest of the leaves talk about production organelles. They also talk about the nucleus and the mitochondria. So it doesn't cover all of the organelles, but it really focuses in on those production type organelles and structures.